Do you ever wonder how scientists can confirm that an organism is a given species beyond its physical appearance? The answer lies within the organism's genetic material, deoxyribose nucleic acid, or DNA. DNA is made up of sequences of building blocks known as nucleotides, each with a base pair of either adenine, thymine, guanine, or cytosine. Each organism has a unique sequence of base pairs. However, most of the time, you can identify a species based on just a short DNA sequence, known as a DNA barcode, which ranges from 400 to 800 base pairs long. These sections contain high genetic variability and are easily amplified, allowing them to be practically examined and used to determine the classification of a species. But how exactly do you use these barcodes to identify the species? You would need to sequence the sample, which would allow you to find the pattern of base pairs within the DNA barcode. This can be done through Sanger sequencing, a technique developed in the 1970s and takes place within a machine. In Sanger sequencing, the sample is first amplified. In normal amplification, normal base pairs, known as dNTPs, are used by enzymes replicating the DNA, giving them the appropriate building blocks to complete identical replication. However, for this amplification, base pairs of dideoxyribonucleotides, which are called ddNTPs, are added as well. These base pairs terminate the chain when added, meaning when used in amplification, they create unevenly length DNA barcode fragments. Each of these fragments has a ddNTP on the end, and each different base pair of these is fluorescently labeled. Next, these fragments are separated by size using capillary gel electrophoresis. DNA barcode fragments are loaded into a gel, which has an electric current run through. DNA is negatively charged and will move towards the positively charged end of the gel, but at different rates. Larger pieces will move slower, and smaller pieces will move faster. After a period of time, the electric current is cut off, leaving the fragments arranged across the gel by size. Following this, a laser is used to identify the fluorescently labeled base pairs of the different chain-terminating bases. The laser first reads the bases in the shorter strands and works its way up to the longer strands. This gives the machine a good read on the position of each base pair in the sequence, which can be used to generate a chromatogram. These figures show the sequence of the base pairs within the DNA barcode. Now that we have the sequence of base pairs, you are able to use online nucleotide databases to determine the species based off data entered from known samples, which is how scientists solve the mystery of the unknown species.